Hello students, welcome to EPG Pathshala. I am Vidya Kothekar. My whole introduction is I am a teacher. I had been at All India Institute of Medical Sciences, JP Institute of Information Technology and Dr. D.Y. Patel Institute of Biotechnology and Bioinformatics. Today, I am going to discuss about a module, amino acids, chirality, peptide bond and polypeptides under the paper Biomolecules and their interactions. As I told, amino acids are the basic building blocks of proteins. All proteins are made up of amino acids. Two different amino acids are linked to each other by a peptide bond. Proteins' chemical properties, chain conformation, 3D structure, function, and specificity all are determined by the properties and sequence of amino acid side chains. So students, let us see what are we going to learn in this module. I am first going to describe the basic building blocks of proteins, the amino acids and their properties. You know, proteins are most important biomolecules. They are structural proteins which are passive, or there are proteins which participate in all the functions of the body, and amino acids are their basic building blocks. I want to discuss next the chirality of the amino acids. Third, I wish to introduce you to the concept of codons for different amino acids. You know, codons connect our proteins to our genes through the sequence of nucleotide controlling the sequence of amino acids, which are called codons. Next, I would like to enumerate the mechanism of peptide bond formation. Lastly, I wish to introduce you to the great field of peptides and polypeptides. Basic building blocks of proteins. Amino acids are basic building blocks of proteins. What we have is a central carbon, asymmetric carbon, which is called C alpha. All amino acids have a backbone of an amine, that is NH2, and a carboxylic COOH functional group attached to the central asymmetric carbon atoms C alpha. With the exception of alpha amino proline, these have a hydrogen atom which is attached to C alpha atom, which accounts for the third valence of C alpha atom. At the fourth position is attached a side chain R. Latter usually has carbon, oxygen, nitrogen, and hydrogen atom. There are few exceptions. Some amino acids, like cysteine, have sulfur, and selenocysteine has selenium. The amino acids differ from each other in their side chain R, that can be either aliphatic aromatic or heterocyclic in nature. Exception is glycine, which is abbreviated as GLY or one letter code G, which has hydrogen atom H attached as a side chain. Amino acids can be classified according to core functional group location as alpha, beta, gamma, and delta based on polarities: positively charged, negatively charged, non-polar, polar. Based on pH level, based on side chain group type: aliphatic, acyclic, aromatic, hydrophobic, presence of S or AC, etc. There is another way to classify amino acids based on sizes. Like class 1 is glycine, very small, no side chain. Class 2, proline, which is special because the chain turns back and connects to backbone NH group. Class 3, A, alanine, S, serine, T, threonine, and C, cysteine, small and polar. Class 4, D, N, E, Q, K, R, and H. These are charged, polar, or hydrophilic amino acids. And class 5 is valine, leucine, isoleucine, tryptophan, phenylalanine, tyrosine, and methionine. These are non-polar or hydrophobic amino acids. We show in this figure the classification of amino acids based on pH levels. Like 21 proteogenic amino acids, that is, those which are normally found in eukaryotic proteins, can be grouped according to their side chain pK values and charges carried at physiological pH 7.4. We show different groups rather. Positively charged amino acids: arginine, histidine, and lysine. Negatively charged amino acids: aspartic acid, glutamic acid. Amino acids which have uncharged chains like serine, threonine, glutamine, asparagine, and special amino acids: cysteine, selenocysteine, glycine, proline. And lastly, a group of hydrophobic amino acids and those having aromatic rings in their side chains. IUPAC nomenclature for the amino acids. The asymmetric carbon is called C alpha. The side chain carbon or oxygen or nitrogen atoms are labeled as per their distances from the C alpha atom. Like the atom next to C alpha will be called C beta. Next to C beta will be called C gamma or O gamma or S gamma, or if there are two atoms, two carbon atoms at the same position, they can be called C gamma 1, C gamma 2. The next two C gamma will be called C delta or S delta or O delta 1, N delta 2, N delta 1, C delta 1, C delta 2, etc. And next to delta will be called N epsilon or C epsilon. If there are two atoms at the same position, then they will be called N epsilon 1, N epsilon 2, or C epsilon 1, C epsilon 2. Next to it will be called C zeta or N zeta. And last, that is N eta 1, N eta 2, etc. We show IUPAC nomenclature for all 20 amino acids. Selenocysteine is not shown here, and you can see the difference rather. Like for example in serine, it is C alpha, C beta and S gamma. Threonine, it is C alpha, next C beta, next C gamma, then O delta 1 and N delta 2. Let us see a long chain arginine. It has C alpha, C beta, C gamma, C delta, N epsilon, C zeta, N eta 1 and eta 2. This is the longest chain. And tryptophan has multiple atoms at the similar position, like C alpha, C beta, C gamma, C delta 2, N delta 1, C epsilon 3, and C epsilon 2, C zeta 2, C zeta 3, and so on. Essential, conditionally essential, and dispensable amino acids. 9 essential amino acids which cannot be synthesized from other compounds are Phe, Val, THR, TRP, MET, Lu, isoleucine, lysine, and histidine. Our body cannot synthesize them, so they have to be given through the food or supplements. 6 amino acids: arginine, cysteine, glycine, glutamine, proline, and tyrosine are conditionally essential. This means their synthesis is limited to a pathophysiological conditions. 5 amino acids: alanine, asparagine, aspartic acid, glutamine, and serine. These are dispensable, which means these are synthesized by our body and one need not take them as supplement. Chirality. Amino acids are chiral molecules. What do we mean by chirality? Molecule is chiral if there is another molecule that is a mirror image of the same molecule. A typical example is of human hand. Your left hand is a mirror image of your right hand. So also are the amino acids. There are left-handed and right-handed. All chiral molecules have a nomenclature. In case of amino acids, these are denoted by D L-cysteine. Both caps generally use smaller size alphabets to designate them. These names have come from Latin words dexter and laevus, and these relate to the structure of glyceraldehyde, which is a chiral molecule. There are various ways to understand the chirality of amino acids. One famous way is CORN, C-O-R-N, CORN notation for L and D amino acids. Look down from H to C alpha atom. The hydrogen atom to C alpha atom, and then look from left to right or in a clockwise sense. Because of the sp3 nature of the C alpha atom, the three groups carboxylic CO, side chain R, and amino group NH. They are equally spaced at 120 degrees each. If you go from left to right or clockwise sense, the L amino acid has groups CO, R, and N. That is why it is called CORN or L amino acid, and we usually have L amino acids in all our proteins. In D form, the notation is just the reverse. It is N, R, CO as we go in a clockwise sense and look from H to C alpha atom. If I show you only a picture of L-alanine and D-alanine, how will you recognize it? There has to be some easier way to recognize the difference between a L and a D-alanine. What is the difference between L-alanine and D-alanine? Just consider yourself holding the carboxylic end of amino acid and standing at one end of the bridge, looking at the other end to a butterfly and the water flowing under the bridge. Now, suppose on your left hand side is the methyl group, then it is L-alanine acid. However, if the methyl group is to your right hand side, then it is a D-alanine acid. Holding a carboxylic end of the amino acid and standing at one end of a bridge with the water flowing under it. Look at a butterfly or something on the other end of the bridge. Consider it to be an amino end. Then, if the methyl group is towards your left hand, as shown in the next slide, it is a L-alanine, and if it is to your right hand side, it is a D-alanine. I show it in the next two pictures. You look at this, the girl who wants to hold the carboxylic end, and look at the butterfly, the amino end, and the methyl group is towards the left hand side, and that's why it is a L-alanine. In the next slide, it is the same, but the methyl group is towards the right hand side of the girl, and that's why it is a D-alanine. Now I want to link your amino acids to your genetic information. Codons for standard amino acids are dependent on the nucleotide sequence on our DNA molecule and code amino acids on our proteins. 20 of the proteogenic amino acids are coded directly on the basis of sequence of nucleotides in DNA molecule. This links our proteins to our genetic code. 3 nucleotides like AAA, AGA, UUC code one amino acid, and these are called codons. The genetic code is degenerate, as there are 64 codons. Because there are 4 nucleotides, first position can be taken by 4, second by 4, and third by 4. So total it is 4 to the power 3 equal to 64 amino acid codons, but they code only 20 amino acids. So our codons are degenerate. We show in this slide codons for 20 amino acids. UUU, UUC both code phenylalanine, whereas UUA, UUG both code leucine. CUU, CUC also code leucine. CUA, CUG also code leucine. Glycine, for example, is coded by GGU, GGC, GGA and GGG, but there are few combinations like UAA, UAG, UGA do not code any particular amino acid, but these are stop codons. Pyrrolysine and selenocysteine, how are these amino acids formed and what codes them? Pyrrolysine and selenocysteine are coded by variant codons. For example, selenocysteine is coded by stop codon UGA seen just now, and there is a special ACCIC element. This ACCIC element is a RNA element around 60 nucleotides with the stem loop structure. Codon tRNA, not found in the nature, can also be used to expand the genetic code. Properties of amino acids. Within hydrophobic group, hydrophobic character shows considerable variation. For example, glycine is less hydrophobic compared to alanine, and alanine less hydrophobic compared to valine, and valine compared to isoleucine. One can estimate the hydrophobicity of an amino acid using delta G, free energy for transfer of amino acid from water to organic solvent. It is called delta G transfer. Can be calculated for different side chains by subtracting the contribution for backbone, that is nothing but the delta G value for glycine, which does not have a side chain. We show in this table delta G transfer values at 25 degrees centigrade for transfer from water to ethanol, for tyrosine, which is minus 9.6, alanine minus 2.1, serine 1.3, threonine minus 1.7, histidine minus 2.1, leucine minus 7.5, methionine minus 5.4, phenylalanine minus 10.5, tryptophan minus 14.2, which is the greatest value because of its size, valine minus 6.3. pK values for amino acids. pK values are defined as the midpoints of the curve at which half of the protons of conjugated acid are dissociated after addition of half or 1.5 base equivalent. There are two different values of pK, pK1 and pK2. One can differentiate between the value pK1 for dissociation of carboxylic group in the acidic pH range and pK2 value for the dissociation of the amino group in the basic pH range. The bifunctional amino acids have an additional pKr value for their side chains. Out of 21 amino acids, only histidine has side chain pK value in the physiological range of 6 to 7.5. We show in this table pK values, that is pK1, pK2 and pKr for the side chain. For glycine, it doesn't have a side chain, it doesn't have a pKr value. Alanine, no pKr value. Leucine, no pKr value. Serine, no pKr value. Threonine, no pKr value. Aspartic acid has pK1 2.09, pK2 9.82 and pKr 3.86. Histidine 1.82 pK1, 9.17 pK2 and pKr 6, which is in the physiological range. Arginine has pK1 2.17, pK2 9.04 and pKr 12.48, highest value. Lysine has pK1 2.18, pK2 8.95 and pKr 10.53. Peptide bond. The peptide bonds are formed between CO and NH groups of adjacent amino acids by the release of water molecule between alpha carboxyl and alpha amino groups. We can see here two amino acids and CO, the oxygen, and NH3, the H2, these two combine and a water molecule goes away. What is left is C double bond O and NH. Because of the partial double bonded nature, they bond to each other, which is called a peptide bond. The peptide bond we show in this picture. Here we see a CO group and NH group. They form a plane. The next amino acid, CO group and NH, they form another plane. And they couple to amino acids. When a third amino acid is coupled, it is called a tripeptide. This can continue for more amino acids, and these are called tetra, penta, octa peptides, etc. Disulfide linkage. Disulfide is usually the end product of oxidation according to the following reaction: 2CH2SH plus half O2 equal to CH2S SCH2 plus H2O. The SH groups in different parts of the structure are thus oxidized to form a SS linkage or a disulfide linkage. It is shown in this picture how two cysteine molecules combine with each other, and SH hydrogen and one oxygen combined go away as water, and a disulfide linkage is formed, which is shown in the lower part of the figure. Disulfide linkage. Disulfide linkage is usually formed by two thiol groups like R, S, S and R, exactly in the same way as peroxide group forms R, O, O, R. It is much weaker bond compared to our normal CC or CN bonds. It is longer. It has a length of 2.1 angstrom. Importance of disulfide linkage. Disulfide bonds play a very important role in folding and stability of some proteins. Usually, these are proteins secreted to the extracellular medium. In general, disulfide bonds are unstable in cytosol with few exceptions. We show in this picture schematic representation of formation of a disulfide linkage between the two cysteine amino acids. What are polypeptides? Peptides are biologically occurring short chain polymers of amino acids. Polypeptide is much longer than a peptide. Polypeptide has generally unbranched peptide chain. Peptides and polypeptides fall under a broad class of biological oligomers and polymers alongside. Polypeptides usually contain 50 or less than 50 amino acids. These are extremely important in many reactions and connect us to the proteins from which they originate. Polypeptides. Polypeptides are distinguished based on their sources as milk peptides, ribosomal peptides, non-ribosomal peptides, peptones, etc. Peptide fragments are used to identify the source of protein. They have many other applications. We show in this picture a portion of a polypeptide chain. Peptides have received great importance in molecular biology for creation of peptide antibodies in animals. This involves synthesis of peptide antibodies against a particular protein of interest, and it's extremely useful in medical research. Peptides are used in many biophysical techniques. These are used in mass spectroscopy, gel digestion, electrophoresis. They are also used for study of protein structure. Peptides and polypeptides can also be used as probes to follow a protein-protein interaction. Inhibitory peptides are very important because these are used for clinical research to examine inhibition of cancer proteins and many other diseases. Their most promising application had been through target LHRH. The students, I would like to summarize what we have learned in today's module. We have studied and understood the basic structure of all amino acids. Then we learned classification of amino acids based on various methods. We studied chemical structure of 21 proteogenic amino acids. We have also learned IUPAC-IUB nomenclature for 20 amino acids occurring in various proteins. We learned what is a chiral protein or chiral amino acids, and we learned what is the L and D forms of different amino acids. We have also seen the mechanism of formation of a peptide linkage. We have described codons for 20 amino acids occurring naturally, and also codons for selenocysteine, which are normally not present. We have enumerated general properties of amino acids, their delta G and their pK, pK1, pK2 and pKr values. We have introduced you to the great field of peptides, polypeptides and their commercial importance. Thank you very much.